Mic test, test, mic test, test.
Hello, Dr. Saira. Can you please confirm? Can you hear me?
Yes, I can hear you. Yeah, I can hear you. Dr. Asma, why your voice is coming like, hello, can you hear me?
Yes, I can. Can't you hear me?
Yeah,
I
now.
just went offline for some times.
Okay.
We are almost close, and as it seems that our presenters are having issues connecting, so it's just me, Dr. Saira, and Dr. Kehkeshan Jabin, and Dr. Asif would be joining us as well. I think so. He's not here yet.
Okay. So what will be the order of presenting? Like,
I think so. We will follow the sequence, and we will play the videos in case if somebody's not showing up. Let me quickly call Vice Chancellor, so he's also available for some maybe opening remarks or something like that. Let me call him.
Okay, I have shared the link.
Dr. Asma?
Yes, Shintaro.
It is time to start.
Yes, and Shintaro, we don't have the other presenters available. They are totally off network, which means I cannot contact them at all.
Okay.
So we will be ready with the presentation of Dr. Kehkeshan, followed by Dr. Haknawaz.
Okay.
and Dr. Mehwish Iqbal. So the starting 3 presentations, you are going to play them. Then Dr. Saira, Dr. Asif, and myself. So how do you want should we go with this sequence, or should we start with the presenters that are available right now? What do you suggest?
Let's start with the people who are connected.
Yeah, that sounds like an excellent plan.
Okay.
So I'm starting right. So, Assalamu alaikum, everybody. Hello and warm welcome and greetings from Pakistan. We at Rawalpindi Medical University we have the privilege of presenting as a team here at APAN58 today. Due to power and unexpected network issues in Pakistan, unfortunately, some of our presenters in the last moment were not be able to join us. But as a backup, we did have, I'm very grateful for my team for staying up late at night, and they did the recordings because it was informed that we are going to have a power breakdown or internet issues in our cities. So they did some recordings, and we are going to play to keep the session rolling in. So that's how we manage as a country, and we are very proud of that. So the sequence of presentations as per the schedule are a little bit deformed. So we are going to be presenting with the presenters that are currently available, and we are keeping our fingers crossed that the remaining presenters will join us as we move on in the session. To begin with, let me introduce about the Rawalpindi Medical University. So Rawalpindi Medical University is a public university located in Rawalpindi, Punjab, Pakistan. It is affiliated with the 4 public sector teaching hospitals, and very famously known as Holy Family Hospital, Benazir Bhutto Hospital, DHU, and so on. And basically, it is for the trainings of undergraduate and postgraduate medical education and trainings. Recently, it has been upgraded to the level of university. It has been operating for many years, for more than a decade, for more than 50 years as a medical college, and then it was upgraded to the university. And we are currently successfully running many PhD programs in different clinical departments. Hopefully, our Vice Chancellor will be joining the session too once he's in the internet, his internet or his connectivity is better. With that, we formally move forward, and I would give the liberty to Dr. Saira Karimi, who is Assistant Professor at the Department of Pathology at Rawalpindi Medical University, to please go ahead with her presentation. Ma'am, will you please take the liberty to start the session with yourself?
Sure. Thank you so much, Dr. Asma, for the opening remarks. I would start by sharing my slide. Thank you so much for having me today and giving me a chance to present as a first speaker representing Rawalpindi Medical University. The formal introduction is, hello everyone, good morning and good afternoon. My name is Dr. Saira Karimi, and I am an assistant professor of pathology at Rawalpindi Medical University. My topic of presentation is the integrated approach of AI surveillance and climate modeling for the airborne allergens in urban environment, which is a very hot topic these days. I'll be discussing an emerging intersection of artificial intelligence and climate modeling, which is focusing specifically on how these technologies, they can be used to predict airborne allergens in urban environment. This topic particularly is increasingly relevant as urbanization and climate change significantly influence allergens distribution and prevalence with direct consequences for the public health. Starting with the introduction to airborne allergens, there is a continuous linkage between environment and an individual's at the time from birth to the death. So airborne allergens, they include a variety of particles such as pollen, mold spores, dust mites, and even pollutants that can trigger the allergic reactions. These allergens, they are particularly problematic in urban areas where environmental factors, they can amplify their presence. And towards the impact of climate change, which is an additional driver of this airborne allergens, which can aggravate the nature of the allergens and also modify them. Here is a representation of some urban gray space areas where there are a lot of surveillance studies that have been done to the different populations to know that each year where there is an industrialization and urbanization going on, what kind of method and samples they have implemented over there, and also what were their findings. So according to this, per each 10% increase in the gray spaces coverage near home, there is 7% for SPT positive, and then there is 10% in allergenic rhinitis and for the polysensitization, and then the list is going on. Urban living, they can be the driving factors for highly prevalence of allergic diseases. Global and local implications. There is a lot of studies there are a lot of reports that the pollution population that suffer from pollen-related allergies, this number is on the rise and then there is an economic burden as well, which is equally concerning. Millions of dollars are spent annually in the countries like European, US, also towards the major part of the European population is under this threat of increasing pollen allergies in the challenge of climate change. The challenge of predicting airborne allergens, because now the world is going towards the climate modeling, and where we can predict that what warmer days we are going to have, what will be the rain patterns in the future, and what could be the nature of those seasonal variations, and what can we implicate on the agricultural side. At the same time, when we talk about the integrated research and multidisciplinary research, these all are interlinked. Environment is linked with the health implications of other human populations. So the challenge of predicting airborne allergens is very difficult. First thing is that airborne allergens, they are really, really complex. There is a complexity of prediction. Why? Because it is not straightforward. There is a lot of parties involved in it. For example, there is aerobiological data where we get the pollen concentration. For example, if we just take the pollen, complexities in, and then there is such a satellite vegetation phenology monitoring, meteorological data is there, and then the current limitation is this, that existing prediction models, they often fall short because they do not fully integrate real-time data or fail to account for the microenvironments within the urban setting. For example, urban heat island, they can lead to localized spikes in the pollen concentration that are not captured by the broader models. When we talk about the challenge for predicting airborne allergens, aerobiological data, then the meteorological data, and then the distribution data, the phenology and flowering data. Then there is a geospatial data. So when there are multifactorial things are involved, then the micro prediction is not possible for a local environment and for the a local geographical data and the geographical population. Here, there is a need for the advanced modeling because the climate modeling as such cannot be implicated on the airborne allergens. So for that thing, we need specific individualistic need for that because the diverse data sets, they are ranging from these all 4 different categories on pollen counts. So temperature and wind speed that are linked with the pollen widespread and the outspread of it. So when we talk about the exposure and when we talk about the environment linked to the human lifestyle and also the human diseases and the prediction of that in the future, there is also a term which is very much into the discussion, which we call as the rise of exposomes. What is exposomes? These are totality of the exposures to which an individual is subjected from conception to death. They refer to a study. It is crucial in understanding how cumulative the exposure to allergens and other environmental factors, they affect the health. And similarly, when we talk about the rise of exposomics, we write everything that a human is exposed to. That could be the increased use of pesticide and herbicide. That could be the leakage of chemical spills over there. That could be the rise of temperature, rise of carbon dioxide, then there are other potential health effects that are associated with these. All exposomes lead to the cancer, vector-borne diseases, fungal and bacterial infections, respiratory illnesses. So in exposomics, what are the importance of exposomes in allergy research? It provides a framework to study how different exposure they interact over time and contribute to allergic disease. When we integrate the exposomics with AI, we can begin to map out individual exposures, profiles, and predict the health outcomes with great precision. So here comes the framework of exposomics. Here in this left side of this slide, there are external exposomes and there are internal exposomes. And then there is health impacts that are interlinked with all these exposomes that are human from the course of its life, from conception to death, is exposed to. Then when we study these exposomics, we take the data collected from it, is detailed, high-dimensional representation of individual health and physiological status. Then we implement the AI and big data and machine learning, deep learning on this kind of data. And then it gives us the, novel and cluster discovery of, it is lead us to the causal analysis. Then comes another another science which is a need of today's challenging world, which is the personalized medicine. How can we cater to the personalized needs of a person who is exposed to all of these exposomics? This is where the AI plays a role in it. Artificial intelligence, it plays a pivotal role in enhancing our ability to monitor and predict the airborne allergens. It can, the algorithms, they can, analyze vast amounts of data from weather patterns to social media mention of the allergy symptoms to provide a real-time surveillance for the allergy levels in the specific geographical population and to the need of a person. So when we talk about the role of artificial intelligence, the case example is this, the use of natural language processing, which is NLP, to extract data from the clinician notes and track allergy trends, track the allergy trends, and then make a data set of it. Another example is deployment of the portable AI devices, because that's what another project which we need to work on. We have seen in the course of pandemic when there was an alert. Mobile alerts were there, and where they confined you to a certain territory where you would not be, allowed to go to a certain after a certain diameter. That's what the future needs from us in tracking the allergy of the patient. So it's the deployment of portable AI-powered devices that can create real-time air quality maps on the one side, and then offering hyper-local predictions. So this provides a framework of from many gaps to be addressed for the adaptation of AI diagnosis, phenotypic, and then there are many allergies which can be tracked onto it. What could be the benefits? The benefits is this, that the AI-driven tools, they can not only improve the prediction accuracy, but also make it easier for the individuals to manage their allergy symptoms. Because whenever there is allergy season at the peak, and when there is not a season that we are going to, because this is what we are predicting from the climate change and from the climate change modeling and the real-time data, that there would be a fluctuation in the season variation. There would be extended summers. We are not having spring and autumn season. So there is a change. So onset of the allergy season and also the extended one, this would be very much easier for the individuals to manage their allergy system if they get the AI-driven tools. Then there is a specific use of AI or ML in environmental science for allergic diseases and asthma. There is an example that what kind of different models they use the data. So there are different pollen count model as well available in the machine learning. Thunderstorm asthma prediction is also there. Wildlife risk is there. Air pollution risk is there in prediction also. This all goes into the machine learning and the big data handling. These are all implemented models that can be used in AI prediction and surveillance for the airborne allergens, and also related to the wild plant and the pollen allergens. When we talk about the severity of responses in patients, because these are the models that can be predicted and then implemented, and the policies can be implemented. What about the response that we get from the patients? Here comes the implementation, and then the response and the track of whatever the model is applied to a specific population. There are some success stories. For example, the success story, one of the model is BioMap82. This gives the prediction of the severity of responses in patients with atopic dermatitis and contact dermatitis, which is exposed to the chemical irritants. So AI is transforming it, because this model aims to advance the personalized medicine, precision medicines, by identifying those genetic biomarkers that can predict the therapeutic responses and disease predictions. This gives a potential clinical laboratory application of AI in allergy immunology. There are some potential application in AI. I will not go into the detail of each one. There are broader application as well. There are clinical applications and there are laboratory applications. In clinical settings, the AI can assist in diagnosing complex allergic conditions by analyzing electronic health records and identifying the patterns that may not be immediately very obvious to clinicians. And then laboratory applications, they can process the larger data set, because they got diagnostic laboratories, they do have larger data sets through neural networks. This is not only will improve the diagnostic accuracy, but also helps in development of safer treatment protocols, because diagnosis is the key. When we talk about, this is how a typical model of AI looks like that, where there's an input layer and there's an output layer, and there are a lot of layers in between that we do in the machine learning. Input layer, again, all the factors of clinical and laboratory, where we get the patient characteristics, reaction history, suspected medication, any history of life-threatening drug hypersensitivity, and then we do, on that data, we do the modeling and expanding the data sources. Then the output layer, if there is a risk of any penicillin-related allergy or something, then we can categorize those, age, gender, or the specific response-specific class of the patients, and then they would be put into the category of high-risk allergy, medium, and low-risk allergy patients. So this is how AI work and can be integrated into the betterment of the humanity. And then there is potential and future applications and directions for the AI. For example, we can expand our data resources. The future of AI in allergen prediction and management, they lies in the expanding the type of data that we collect, where data collection procedures might need to change from real-time environmental senses to patient-reported outcomes via mobile apps. That's what we have discussed now. Advances in AI models also, such as deep learning and supervised learning, will enable us so that we can better identify the relevant inputs like pollen identification and map these to health outcomes with great accuracy. Of course, this is not possible without the collaboration of multi-sectorial sectors, multi-sectorial dimensions, across the disciplines. Only not biology or health sciences. We need everyone together on this platform, combining the expertise from the environmental sciences, AI, and medicine. That would be the key to unlocking the full potential of these technologies in improving the public health. And this is the take-home message, that let's all work together. At the end, I would like to give a big thanks and shout out to APAN that gave us the opportunity to present. Rawalpindi Medical University, giving me a chance for this recognition, and last but not the least, Higher Education Commission, which always thrive for its faculty and the population. Thank you so much. This is all from my side.
Thank you. Thank you so much, Dr. Very, very insightful. And yes, as the world has entered into the healthcare 4.0, so we are moving towards the use of AI in different fields, including the field of medicine. And it comes as no surprise that AI tools and AI usage in the detection and the prediction of the allergens is going to be the future. And we are so happy and so glad that the medical universities have taken up and amalgamated and are working in collaboration with these different techniques. So that was a great talk. Moving towards our next presenter, we have here with us is Dr. Asif Maqsood Butt from Rawalpindi Medical University. Dr. Asif Maqsood Butt is also the Manager of Research, Innovation and Commercialization at Rawalpindi Medical University, and he'll be talking about the role of e-tech in improving healthcare communication and quality adjusted life. Yours, Pakistan Roadmap to Achieve Sustainable Developmental Goals of 2013. So here from RMU, you are going to see a lot of tech-based presentations today. Dr. Saira Karimi, do you have something to say?
Yes, our Vice Chancellor wants to join, but for some reason he's not able to join. Can you please check? He just gave me a text.
Shintaro, can you please check if is there any issue? Because I think so I have shared the correct link with him.
If he has the link, he should be able to connect, but I don't see anything from his side.
I'll share the link again.
Okay, thank you. While we wait, maybe we should move to Dr. Butt's presentation because Dr. Asma has frozen.
Already.
Yes.
Assalamualaikum. In Urdu, it is in Pakistani, greetings. So it's really honored and privileged to me, being as a faculty member of Rawalpindi Medical University and from Pakistan, to present in this prestigious forum. And I'm highly honored and thankful for APAN conference for allowing me to present my innovation model. So here, let me share the screen. Is it available? Can you see that?
Please put it into slideshow.
Yeah, this one. Is it fine now?
Yes, we see the slide. Health
Okay,
status.
thank you. Yeah, thank you so much. So without wasting the time, the role of e-tech in improving quality adjusted life is in Pakistan. I'm Dr. Asma, but I'm public health specialist and innovation expert. Currently, I'm doing my PhD in Health Service Academy. And this is a small brief introduction of myself. And I'm also the public health lead for Pakistan from China-Pakistan Medical Association. And we are promoting the One Belt, One Health Corridor and strengthening the healthcare system of Pakistan by interacting with the bigger institutes, universities in Pakistan and the healthcare system by improving and introducing the integrated healthcare services and innovative models, the augmented virtual reality and other aspects that improve the innovation quality in Pakistan. So regarding the health status, if you see the statistics, we can analyze that the number of the infrastructure, the health facilities, the health sources in Pakistan are very compromised. And from 10 years, we analyze the data, which are very minutely changes have been seen. And one doctor is available for more than 700 people, which is for the normal preventive healthcare services, and for the specialist services, more than 6,000 people are, getting the one doctor, specialist doctor services. Moving towards the comparison of the indicators, which are very alarming situation. If we compare the 2019-2020, we can see that there are majority of the indicators which are predicting the life quality, which are very minutely changed and seen as the stagnancy in the quality of care provision in Pakistan. Similar, the data can be visualized here in 2023 and 2024. We've seen that the majority of the health indicators, the mortality, the maternal health, and the birth rate, and the prevalence of the infectious diseases is very alarming. And the coverage, that is the immunization and other coverages in the preventive healthcare, is not seen as improvement, which should be, in last 10 years. So for the improvement and achieving the sustainable development goals, it seems very, very slow. Then we can, moving towards the capacity building, we see that the infectious disease prevalence and, the training of the staff, which is only 26% of the individuals seen that they are getting the training, and 64% had put all the responsibility on administration, that they are not giving the right platform to, putting their capacity building, workshops or trainings, as 74%, then they are never attended any of the training. This research we have conducted in last few years in tertiary care and secondary care hospitals. And similarly, another study we conducted in Pakistan, which is about the specialty choices among the future doctors of Rawalpindi Medical College that time. So in perspective of public health, so we have come to very alarming situations in the healthcare delivery system and services, that the referral system is very compromised. The primary healthcare services are low, achieved by the people in the community there, and the majority of the people are seeking healthcare in the private sectors, and the follow-up and compliance is also like they are hooping around, and this leads to the resistance and chronicity in the in community. The role of the family physician is very important in Pakistan, because 60% of the population that approach family physician or the GPs and the private clinics and hospitals, but the research level at that in this category is very low, and there is a very low social mobilization in this sector, and so this leads to very poor disease surveillance and data collection, which leads to many questions. And this also puts alarming situation for disease control and outbreak control in Pakistan. If we compare if we see the state of the affair in the public health sectors, which shows that less than 2 minutes are being given to any patient, because the poor referral system leads a patient coming to the tertiary healthcare hospitals, and there is a huge burden on the tertiary care setups. So as compared to other countries which are giving good time for the patient care and for the diagnostic services, and this is the public health care situation. But in the private setups, a good time is being given to the patient care for their history and examination and diagnosis services. And then how we can improve the role of the doctor and role of the public health? This needs a community engagement. So community engagement needs the health-seeking behavior to be improved in the primary healthcare level and the secondary care level in the areas where people are living, and we need to improve the health-seeking behavior as well as the health risk belief in the community, so they can adopt and opt the life which is leads to a quality adjusted life to improve. So other thing is that the doctor level that must be improving the primordial, preventive and promotive healthcare services, that is the health education to be improved at all the sectors of the healthcare services. And also the rational treatment, the curative treatment should be given, and that's how we need to be addressed, that there should be a very rational treatment of the antibiotics, the injectables, that all should be addressed. And in the recently, which is not as been practiced. Similarly, rehabilitation services also need to be look on this for a proper solution, for proper healthcare services, for rehabilitation services, vocational and psychological and physical rehabilitation services need to be improved with a preventive healthcare aspect including in that services. And so we can look on the SDGs. These all are the aspects which are need to be addressed in a integrated way. So the gap we have found in all these setups, in all these sectors, sections, we found that there is a lack of the integration, coordination, and synchronization in all the categories of primary to tertiary healthcare services, even the institutional levels, even in the diagnostic levels. So there are this lead us to provide the national health policy for integration healthcare system, and that is because that is the roadmap to achieve the sustainable development goals. One of the paper I have published in the Rawalpindi Medical Journal, which is the proposing solution for the integration level in the public health sector, same as we are proposing for a similar setup for the private healthcare sector, which should be in a primary, secondary, and tertiary healthcare levels, and that should be inculcated with the integrated with the certificate of public health, so so that our reference system could be maintained same in the primary public health sector and similar in the private healthcare sector. So this will improve and give the domain and the authority to a UCMO, the area in charge, for social mobilization, for integration to the community, and screening level should be improved. This will also improve the surveillance of the area, and this will lead a research, and that research will improve the evidence-based data for improvement of the healthcare status, to reduce the resistance and chronicity in their health in their community. So Pakistan quality adjusted life years to improve and reduce the disability adjusted life years needs and all the levels improvement and integration that a patient should feel like honored, and the doctor and patient relationship and the doctor and patient interaction should be well addressed. And that's how we are improving, that's how we are educating that in the community, the preventing diseases, the promoting health and prolonging life. This needs all the levels integration and coordination, and all the levels should be well managed, and there must be inculcated with all the sectors, and there must be a reduce in the over-the-counter medications, and all the community-driven healthcare services must be low down, and the healthcare services from the government, from the authorities should be addressed in a right way. So one of the article I have presented in 2020, that is health for all. So how we can achieve the health for all when we synchronize the public and private care using e-tech to improve care, healthcare in the healthcare delivery system. This is the locked variant because this is the innovation model, and this model have three more components, which are having the pre-post and epidemic community communication services and strategic response system, as well as the MATRIS, the medical aid teaching and rescue system, the rescue support system, which will be which are in the patent services and which will be ready in next around 1 year. And we are very well confident that this speech and this presentation will motivate the stakeholders, the authorities, the policymakers to help us out for implementation of all the services in Pakistan for improving the quality adjusted life years in Pakistan. This model also contains few of the other segments which we are not disclosing as the locked patent variant. So, but when the authorities will contact, we will be honored to address and pitch our model to them. So to build a happy Pakistan, the happy mean health education, promotion, and innovation in Pakistan needs integration, levels to improve, communication to strengthen up, and reporting system in Pakistan should be well addressed. And this is all from my side. And these are a few glimpses of our previous achievement we have achieved for Rawalpindi Medical University and for Pakistan. And we have achieved 2 gold medals, Best Achievement Award and bronze medal for Rawalpindi Medical University and Pakistan. This is all from my side. I am honored and thankful to my Vice Chancellor of Rawalpindi Medical University to mentoring in such a way that we are now, leading in front of the globe. And Pakistan is, is improving for digitalization and strengthening the healthcare system. Thank you.
Thank you, Dr. Asif. It was an insightful presentation. Thank you so much. It is indeed a great honor to represent Rawalpindi Medical University and show your achievements so far. Moving on towards our next presentation, I think we are going towards the recorded ones. Shintaro, are you there with me?
Yes, I am.
Okay, so I would like to request you to, can you please play the recorded video of Dr. Asma Umar Khayyam, because she's having some network issues. So we're gonna have the lecture, recorded lecture of her.
Okay.
Thank you.
Hello, everybody. Welcome to APAN58, a presentation from RMU. So this is Dr. Asma Umar Khayyam. I am assistant professor at the Department of Biochemistry at Rawalpindi Medical University in Pakistan. And I have the privilege of presenting at APAN58 today. So my talk or my presentation will be is titled as Data-Driven Strategies for Hepatitis C Management, Lessons from Clinical Analysis in a Pakistani Setting from the year of 2013 to 2024. Introduction is my research or my presentation today is about the hepatitis C. As we know, it is one of the major public health issues worldwide, globally, and particularly in Pakistan. It is estimated that approximately 11.5% prevalence of HCV infection in the adult population in the Pakistan. So there is a major public health issue regarding hepatitis C virus in Pakistan, and Pakistan ranks globally in terms of hepatitis C virus infections, with approximately 1 in every 20th Pakistani is reported to have already been infected with it. And another alarming statistics that I would like to share is the mortality rate due to the HCV currently at Pakistan. It stands at around 366,000 annually, which is quite big. So this slide shows the prevalence of the HCV infection in different provinces of Pakistan. As you can see, the provinces here, you can see there's Punjab, Sindh, Khyber Pakhtunkhwa, Balochistan, and tribal areas. So the literature reports that the majority of the cases are reported from the tribes of or the province of Balochistan, and followed by Khyber Pakhtunkhwa and Punjab. And the risk factors, or the most common possible risk factors associated with the transmission of the HCV infection in Pakistan, are reported to be predominantly like mishandling, specifically like shared needles amongst the drug users, and unsafe shaving practices or unsafe barber practices. Also, the hospital waste, it is considered to be as a major risk factor. HCV infected hospital waste is not properly disposed. As a result, it too is contributing as a major risk factor in the transmission of the HCV infection in Pakistan. Alongside surgical instruments, not proper sterilization of them is considered to be as one of the risk factor contribution, as well as tattooing needles. Using unsafe or unsterilized needles for the tattooing is considered as one of the risk factors, along with the spousal contact with the deceased HCV patient. That is also known to be one of the risk factors associated with the HCV transfection here in Pakistan. Now, my study context is, we know that the current challenges that this disease load is here in Pakistan is tremendous. Now, this disease is basically a very complex disease, and it has an interplay of various hematological, hepatic, and radiological factors. And understanding these factors is very important for improved diagnosis and treatment, particularly related to the HCV virus. Now, the current study objective of my study is to leverage advanced predictive analytic methods to enhance the management and treatment outcomes of the hepatitis C among patients at Rawalpindi Medical University and its allied hospitals. Now, the materials and methods that I used for my study my study was for over a period from this study period was from the 2013 to 2024, and the study population that I used for my study was a retrospective analysis of around 9,392 patients that visited the Holy Family Hospital, which is an allied hospital of the Rawalpindi Medical University, with the suspicion of HCV infection. So around 9,392 patients, we used retrospectively analysis of these patients for our study. Now, how this data was collected? So as you know, here in Pakistan, a majority of our medical record keeping is done on pages or on the paper. So basically, we use those files retrospectively, we retrieved those data, and then we digitally managed or entered all those into these different softwares for data analysis. So here, this slide would be showing you how we collected manually, how these data was recorded in different files and properly maintained at RMU, and then that was digitally transformed or digitally entered. The data was entered. In here is, you can see the Excel sheet where we've entered the data, and then it was moved forward for different analysis. So if we are to discuss what the patient data or the recorded parameters that we were using, so this slide would show that. So the different categories that we had used for our data was we basically record it's a standard method of data recording for the HCV. So we do demographic information recording, then we go for laboratory test results. They are recorded. Molecular molecular diagnostic tests, they are also recorded, and the diagnostic images are also included in our routine recording. And the parameters, if I am to describe, as you can see here, there's a category, then you can see there is this parameter, and there is this description. The description is for those who are from the non-medical background. So the demographic information, we basically record or we used age, sex, and location of the patient. The laboratory test was we used the hematologic data. Then we used the LFTs, which is the liver functioning test. Then we recorded the renal functioning test. So the hematological data, we use the complete blood count, hemoglobin, TLC, which is the total leukocyte count, and platelets. And for the liver functioning test, the recorded, the retrospectively data that was retrieved, it included the bilirubin, the alanine aminotransferase, which is ALT, the AST, aspartate aminotransferase enzyme, ALP, alanine phosphate. So these were the LFTs. And for the renal functioning test or the RFTs, we recorded urea and creatinine. The molecular diagnostic test, which is the PCR of the HCV virus RNA, basically it is used for the quantification of the RNA in the blood as per the international standards. So we can know the viral load of the HCV in the patient. That is also routinely recorded. And then we also recorded the diagnostic images, which was the abdominal ultrasound. So basically this imaging will show the abdominal organs, including the liver, to show that we can have a clear picture of the disease progression and the current state. So this is the patient data that we used manually from the papers. We entered these data into the different softwares, and then it was moved forward towards the other analysis. So the other advanced analysis were the predictive analytics. So the predictive analytics, we use this retrospective data to do the predictive analytics. What we used was, I used the statistical method, which was the logistic regression. Then we also used this decision trees and cluster analysis. So basically these analysis, they collectively offer a comprehensive review of how biological parameters they impact the HCV management and the patient outcomes. And now they will provide valuable insight for improving the treatment strategies at RMU and its allied hospitals. So here is a flow chart of how I went about with my data analysis. So basically predictive analytics, in predictive analytics, we use the statistical techniques, or we can use the machine learning and data mining. So what we do here is, using the predictive analytics, we analyze the historical or the retrospective data and the current data, and using that data and using these different tools and techniques, we can forecast the future events. So here we did the predictive analytics of 3,092 HCV patient data that I showed you in the previous slides of how we retrospectively retrieved them. These data were the data was collected, then the data went under data cleaning, and then the feature selection. We decided which are the parameters that we are going to study in relationship. So here in the second step, it was data collection and preparation. In the third step, we went towards the model building. So that is here the predictive model building. So we used the different methods. The statistical method, first of all, we used. For that, we did the regression analysis as described previously, and we are going to go in detail about why and how I selected each of that. Machine learning, for that we did the algorithm, decision trees and cluster analysis, then we did the prediction and evaluation, and then finally we made the data-driven strategies. Now the analysis for the logistic regression is basically it predicts the likelihood of achieving a sustained SVR response and the risk of advanced liver diseases. The data or the parameters that we studied was ALT, AST, bilirubin, and HCV RNA levels. And based on this model, we did some prediction, and the results showed that the higher HCV RNA levels elevated the ALT, AST levels and decreased the SVR likelihood. Similarly, lower bilirubin levels are positively correlated with the SVR. Advanced liver disease, high ALT, AST levels, and elevated bilirubins are significant predictors. They were known to found to be significant predictors. Similarly, this model provided probabilities for progression to cirrhosis, aiding in early intervention. The next, which was the machine learning tool that we used, the decision trees. Basically, the objective was to classify the patients based on their risk of severe liver disease and treatment response. And the data that we used was CBC, LFTs, PCR, and ultrasound. These, the data was used to build the model. So we created the decision trees to establish clear rules for classifying the patients, correlating it also with the clinical data that was present, as well as the literature. And the results was the disease severe classification. We could successfully do that. So identified thresholds for biochemical parameters that classify patients into risk categories for severe liver disease. Also, elevated AST and bilirubin levels were found to be critical for identifying high risk of cirrhosis. The third we did was cluster analysis, and our objectives was to group the patient with similar biochemical profiles into clusters. And the data that we used was CBC, LFT, and PCR ultrasounds. And the clustering patient into identify patient into the subgroups. And as a result, we could identification of subgroups could be done. We revealed distinct patient subgroups with unique biochemical profiles. For example, one cluster had a high ALT and HCV, but low bilirubin. Another had the opposite. So we could basically, based on these results, we could classify them. So, the classification would result in better tailored management, suggested different management strategies based on the clusters. So high HCV RNA and high ALT cluster require more intensive treatment and closer monitoring. So from these tools, we drew some strategies for the HCV management. So the strategy was basically, you can see in this table, I have formulated or tried to compile it. So the strategy is basically strategy, description, implementation steps, and the expected outcome. So risk strategy stratification, we did predictive analytics for treatment optimization, resource allocation and cost effectiveness, public health campaigns and education, implementation of disease support tools, and continuous monitoring. So the expected outcome of these are like for the risk stratification, we could have improved targeting or intervention for personalized treatment. For predictive analytics, improved treatment outcomes, we can have more personalized treatment regimes. Resource allocation would be helpful in optimizing the use of resources and reduce the basically overall cost of that. Public health campaign and education, obviously it is going to help in increasing the awareness. Implementation of the disease support, using these tools, decision support tools will help in enhanced clinical decision making and improving patient management. Continuous monitoring and improvement strategy will help the expected outcome on ongoing improvement of management practices and better patient outcomes. With this, I would like to close my presentation, and this is some of the data that we used during the analysis. Due to the time limitation, I could not go into the details, so I tried to summarize my study as much as I could. So these are some of the references, and thank you so much for your patience, and thank you so much for listening to me.
Thank you so much, Dr. Asma. Would you like to say something?
Yes. So as expected, I was expecting that I would be off the network. So during my presentation, I could not. But smart enough of me that we had a very good recording of the session previous on late at night. Thanks to you, Dr. Saira Karimi, and the others who had guided on that. So yes, unfortunately, I joined it towards the end of my presentation, but I think so it went well. And if there are any questions, either they can, due to the time limitations, I would like to take them either in my inbox or directly on my email address that has been shared with the team. So with that, Dr. Saira Karimi, I would like to request you to please moderate the session further on because the internet here with me is unstable.
Okay.
Over to
Thank you so
Dr.
much, Dr. Asma, for a thought-provoking presentation. Your contribution to the field are invaluable, and also your expertise and passion for the subject are truly inspiring. I would like to, we are honored enough that with us here, we do have our respected Vice Chancellor. I am honored to introduce our esteemed guest, the Vice Chancellor of Rawalpindi Medical University, Professor Mohammed Umar. Sir, if you are here, can you please open up your camera? And we would like to have a closing remarks or some remarks from your side so that then we can continue with our session.
Bismillah ar-Rahman ar-Rahim. Can you hear me?
Yes, sir, we can hear you. Can you please turn on your camera?
So I can't because I'm somewhere which is difficult for me. I can, but I hope you can hear me.
Yes, sir, we can hear you. Can you please continue?
Okay, so,
Thank you.
Yes, so thank you very much for the invitation and, very sort of exciting session. I was listening throughout Dr. Asif and Dr. Khayyam, and I think these are two excellent presentations, and particularly Dr. Sara has first time I've seen this in details. Although I'm part of this study, but I think the conclusion she has made that this is the evidence-based approach of almost 10,000 patients. I think this is the biggest data ever used for at least in the allied hospitals to make to stratify the patient, to decide about the severity of the disease and cost effectiveness and resource management, and prioritizing the treatment guidelines, particularly in low-income countries. So I think that's wonderful. My third comment is we should follow up this study and contact to these patients because I can see some of the patients have hepatitis from last 1 decade or 2 decades. So if we have a follow-up of these patients in the form of outcomes, which may be in the form of full recovery, or it may be hepatocellular carcinoma, or it may be decompensated liver disease, so we can further formulate what will be the incident and risk of this population, this cohort of developing complication. And we can screen this patient earlier and we can prevent the further complication in this cohort in the form of hepatocellular carcinoma and, decompensated liver disease and liver transplant, as we were discussing on yesterday. So I think this is excellent data and excellent presentation, and congratulations. Thank you very much.
Thank you so much, sir, for your enlightening comments, and your leadership and dedication to the medical education have inspired us all. So thank you so much for joining us. Moving towards our next presenter, we do have with us Dr. Kehkashan. Dr. Kehkashan, she's an assistant professor at Rawalpindi Medical University, and she's going to present her talk, and she's going to discuss the unrevealing hepatitis B, mitochondrial dynamics, immune modulation, and novel therapeutic pathways. Dr. Kehkashan, can you hear me? Dr. Kehkashan, are you here? Please unmute yourself.
Assalamu alaikum, Dr. Saira. I am here. I can hear you. So I request you just play my recorded video.
Okay. Okay. All right. Shintaro, are you here with me? Can we please continue with the...
I am Dr. Kehkashan Jabin from Rawalpindi Medical University, and the topic of my presentation is Unrevealing Hepatitis B, Mitochondrial Dynamics, Immune Modulation. HPV infection is characterized by alteration of mitochondrial dynamics, which causes damage to the organelle by depolarizing its membrane, producing the reacting oxygen species, disturbing calcium homeostasis. Mitochondrial damage and oxidative stress are the main events leading to chronic liver disease, which is progressing from hepatitis to fibrosis, cirrhosis, and finally to the hepatocellular carcinoma. Mitochondrial dynamics plays significant part in the apoptosis process, and it undergoes two opposing events of the fission and fusion. From here you are able to see when there is HBV infection, what happened. There is phosphorylation of the DRP1, which is a mitochondrial damage protein. What will happen after its phosphorylation? There will be the fission of the mitochondria. You know that normal morphology of the mitochondria is tubular. So after the fission process, it gets fragmented. And in this study, we will see how we will use the antioxidant and calcium modulator to convert or partially restore this fragmented mitochondria to the normal tubular form of the mitochondria by the process of the fusion. For it, the hypothesis is just to evaluate the effect of the oxidative stress and calcium deregulation on mitochondrial damage, FAM, which is a host factor, interleukin 15 and 11 expression during the HBV infection. And for this purpose, our main objective are to investigate the role of the oxidative stress and calcium deregulation in causing mitochondrial damage in HBV expressing cell, to evaluate the protein expression of the different mitochondrial fission protein, which is DRP and phospho-DRP1 involved in mitochondrial damage in HBV expressing cell, and to also examine the differential expression paradigm of the cytokine and host factor in vitro and in vivo and their potential role in inhibiting the HBV replication. For first two objective, what was the strategy? We performed immunofluorescence confocal microscopy. We used the two type of cells, Happy D38 cell and HepG2 cell. HepG2 cell were transfected with the plasmid HBV 1.3 mer. Then before the fixation, we treated it with the 50 nanometer micro tracker red. Before the cell fixation, and protein profiling is done by the Western blotting, while the transfection efficiency is measured by the fluorescence microscopy. So we check the effect of the four compound. One antioxidant, that is N-acetylcysteine, and the three calcium modulator, which are EGTA, BAPTA-AM, and the Rule 360. So first of all, we are checking the effect of the NAC treatment. There are three rows. This is a negative control, this is a positive control, and this is treated with the NAC. You are able to see in the negative control there is no core protein. In the positive control, core protein is present. And from here, when there is no HBV infection, the mitochondria is in the tubular form. After infection, it gets fragmented. But when it is treated with the NAC, you are able to see there is the partial restoration of the tubular mitochondria. In the Happy D38 cell as well as in the HepG2 cell, you are able to see here after treatment, there is partial restoration of the tubular mitochondria. Then we check the fission protein. First of all, we did the control experiment and we checked the phosphorylation of the DRP1. You are able to see from the graph when there is no HBV infection, there is less phosphorylation, and when there is HBV infection, there is increased phosphorylation, and we find the significant result in this case. Then we check the effect of the NAC treatment. You are able to see after treatment with the NAC, there is down regulation of the phospho-DRP1, which means that the mitochondrial damage is partially restored. And in the other, you are able to see there is down regulation of the phospho-DRP1. Then in the immunofluorescence microscopy, we check the effect of the EGTA again, negative, positive control, and after treatment with EGTA, you are able to see there is some swollen mitochondria or there is a partial restoration of the mitochondria in HepD as well as in the HepG2 cell. Then we check the EGTA effect on DRP1 and phospho-DRP1. From here, you are able to see when it is treated with the EGTA, there is down regulation of the DRP1 and phospho-DRP1 protein. Then BAPTA treatment, negative, positive control, and after treatment, there is partial restoration of the mitochondria. And here again, in the HepG2 cell, there is partial restoration of the mitochondria. And in the DRP1 and phospho-DRP1, there is down regulation of the phospho-DRP1. The effect of Rule 360, negative control, positive control, and after treatment, there is partial restoration of the mitochondria in the HepD38 as well as in the HepG2 cell. And same is in the case of phospho-DRP and DRP1. After treatment with the Rule 360, there is down regulation of the phospho-DRP1. So for the second one objective to examine the differential expression paradigm of the cytokine and the host factor in vitro and in vivo, their potential role in inhibiting the HBV replication, the methodology was we again use the two type of cells, HepD30 cells and HepG2 cell. HepG2 cell was transfected with the plasmid HBV 1.3 mer. We checked the FAM and IRF3 protein by Western blotting and interferon beta, FAM 26F, interleukin 11 and interleukin 15. We check their mRNA expression, and in the different patient group we check FAM, interleukin 11 and interleukin 15. And the statistical analysis was performed by using the GraphPad Prism. We performed t-test and one-way ANOVA to check the significant values. We check the expression of the IRF3, FAM or interferon beta expression after the HBV infection, and you are able to see from all the graph in both the cell line there is down regulation of all the protein after the HBV infection. And when these all protein treated with the NAC, you are able to see there is up regulation of all the proteins and mRNA expression of these protein, which shows there is the restoration or partial restoration of these protein after treatment with NAC. Similarly, with treatment with the EGTA, there is up regulation of all the proteins and mRNA expression. With the BAPTA treatment, again the same trend, there is upregulation after the treatment. After Rule 360 treatment, again the trend is same. There is upregulation of all the proteins and mRNA expression. And then this same thing we checked in the patient, HBV patient. We divide them into 4 groups: control, inactive carrier, recovered cases, and chronic HBV. You are able to see there is a upregulation of FAM in the control. When there is HBV infection, it's downregulated. In the recovered, it's upregulated, which is again support our in vitro experiment. In the case of interleukin 15 and 11 mRNA expression, when there is HBV infection, there is significant downregulation. And when it is treated with the NAC, EGTA, BAPTA or Rule 360, you are able to see the interleukin 15 expression increases after NAC, EGTA, BAPTA or Rule 360 treatment, which is again showing the restoration. And interleukin 11 is also after treatment with NAC, EGTA, BAPTA and Rule 360 shows the upregulation, means again the restoration of the interleukin. And we also check the interleukin 15-11 expression in the HBV patient, in the groups, HBV patient group, inactive, recovered and chronic. After the infection, there is down regulation in both of it, and in the recovered cases, it's increases, which further support our in vitro experiment. So overall, the result of the study positively indicate towards the positive effect of the antioxidant and calcium modulator on the HBV-induced mitochondrial damage, and the future prospects are the further drugs as well as the drug target against the various proteins of the mitochondria and calcium homeostatic pathway, ultimately preventing the mitochondrial damage, can be explored. And our study is the first to show the association of FAM with the HBV infection, and it is proposed that the FAM expression could be an early predictive marker for the HBV infection, so it's worthy for the further investigation. So this is all from my side, and in the end I would like to acknowledge my supervisor, Dr. Shaheen, Dr. Anila, Dr. Aleem, and the Vice Chancellor of the Rawalpindi Medical University, Dr. Umar, for giving me this chance to present my work. And thank you everyone who were listening to me. Thank you very much.
Thank you, Dr. Kakasha, for the wonderful presentation. And also it was a really detailed one and you presented really well. Thank you so much. Moving towards our the next speaker, Dr. Mehwish. I would request Tara to please play her video, recorded video.
And welcome to my presentation. I'm Mehwish Iqbal and I'm working as an assistant professor at Rawalpindi Medical University. So today we will be talking about 130, that is a chromatin remodeler, and we will try to investigate its role in DNA repair. So the outline for today's presentation will be: first I will introduce you about the topic, we'll talk about aims, we'll discuss the results, conclusions, and at the end we will talk about future directions. So next, 130 is a chromatin remodeler. So in 2009, the lab from which I have done my PhD has shown that 130 has chromatin remodeling activities. They have shown that 130 is able to slide the nucleosomes from one position to another position. 130 can also transfer histone dimer from one position to another position, as well as it can transfer the whole histone octamer. So why we worked on yeast? So because there are many similarities. First of all, we will talk about its orthologs. So 130 is its name in budding yeast. It has its orthologs in other species, like in fish and yeast, it's F53. In mouse, it's ETL1, and in human, it's MARCAD1. So there are many similarities between yeast and human. Like human, yeast is a eukaryotic organism. It contains nucleus, and its DNA is packed into chromatin like human cell. It's single cell, it can be easily grown, and it's easy to manipulate yeast genetically. There are many disease-causing proteins in human that do have their counterparts in yeast. Also, many of the DNA processes, including the DNA repair, is common in both human and yeast. So we will discuss a little bit about DNA damage. So we all know that there could be many reasons of the DNA damage. So depending on the kind of damage, there could be different kinds or the types of the damage induced in the DNA. So one of the most cytotoxic form of the DNA damage is DNA double-strand breaks. So it's a damage when both the strands of DNA got nicked at the same time. So cell is a smart, nature, evolution is smart. So cell has evolved different repair pathways in order to repair this damage. So one of the pathway is called as DNA is called homologous recombination. So homologous recombination is a pathway to repair the breaks that are double strand breaks. So once a double strand break is induced, one of the initial factor that is recruited at the break site is called as MRX complex. So then there will be exo-1 exonuclease. It is recruited and it chops the DNA, which means it is initiating or it's doing the 5' to 3' DNA intersection. So as a result, there will be DNA overhangs, then there will be RPA proteins will be recruited, And these overhangs will look for the homology within the sister chromatids in order to repair the damage. So next, in 2012, two back-to-back Nature publications have shown that FUN30 has a role in DNA repair. They have shown that FUN30 is physically recruited at the damaged sites where it helps in the DNA intersection. As a result, it is favoring the homologous recombination pathway of double-strand break repair. So the aims of today's presentation or today's research work was to investigate the interacting partners of FUN30 to study the role of FUN30 in DNA end resection. So in order to achieve the first objective, that was to investigate the interacting partner, we have pulled down the tap-tagged FUN30. So tap-tag composed of a protein A part, a TEV protease cleavage site, a calmodulin binding peptide, and a bait protein. So it's a two-column affinity purification method. In the first affinity column, it's the IgG beads in which our protein of interest will bind to the IgG beads. In second affinity purification, our protein of interest will bind to the calmodulin beads. So protein A has affinity to bind the IgG beads. Then we have added the TEV protease cleavage. TEV protease cleavage, it will make a cleavage at this region. As a result, this calmodulin binding peptide will be exposed and our protein of interest will bind to the second affinity purification column, that is calmodulin beads. So from these beads, we will elute the protein of interest. So this is the western blot and silver strain of the eluted protein. You can see that we have done the purification at lower salt stringency and higher salt stringency. So you can see that in higher salt stringency, there are many less number of the proteins compared to the lower salt one. In higher salt, there are very less proteins. So we have sent these proteins for the mass spectrometry analysis, and our mass spectrometry data reveals that FUN30 interacts with several of the nuclear membrane proteins. So you can see that NSP1, NUP60, NUP2, NUP57, many proteins that belong to the nuclear periphery, they were pulled down. So this means that FUN30 might be doing something at the nuclear periphery. So research has shown that the persistent double-strand breaks, they are recruited or relocated at the nuclear periphery, where some alternative repair pathways help them to fix the damage. So this means that FUN30 might be playing a role in relocating these persistent double-strand breaks to the nuclear periphery, or interacting with these persistent breaks at the nuclear periphery. This still needed to be explored. So next we have done the co-immunoprecipitation and find out that FUN30 is physically interacting with NSP1. You can see that we have done the MIC pull down, and we got the NSP1 in the MIC pull down, which means that FUN30 is interacting with NSP1. That is also a nuclear pore protein. Investigating the co-localization of FUN30 with PUS1, that is a nuclear protein. So the purpose was to find out the exact location of the FUN30. So in order to investigate this, we have used a split GFP technique in which GFP functional molecule is split into two halves, GFP11 and GFP1 to 10. So these two do not flourish individually, but they can flourish once they are expressed in the same cellular compartment. So we have tagged GFP11 with PUS1, whereas FUN30 was tagged with GFP1 to 10. So you can see that when GFP11 and PUS1 expressed, only the fluorescence was observed in the red channel, that is M cherry. No fluorescence was observed in the green channel. When FUN30, that is the GFP1 to 10, was expressed alone, no fluorescence was observed in red or green channel. Whereas when both of these are expressed in the same cellular compartment, we have observed the fluorescence in red as well as in green channel, which means that FUN30 is co-localizing with the PUS1, that is the nuclear protein. So next we will move to the other objective, that is to study the role of FUN30 in relocation of persistent breaks towards the nuclear periphery. So in order to achieve this, we have done the co-immunoprecipitation. So we have used the cells that were expressing MIC-tagged FUN30. So this is the process of CHIP. We cross-link the proteins to DNA using the formaldehyde. Enzymic antibody was added. Protein D-magnetic beads were then added. And bound proteins, they were washed, these decross-linked in order to release the DNA, and then specific DNA was detected using the primers that were specific, and then quantification was done. So our results have revealed that FUN30 is also important for short-range DNA and resection. So previous studies have shown that FUN30 is important for long-range DNA and resection, whereas in this study we have shown that FUN30 is able to favor the resection even for the short range. So this is the quantification of DNA and resection that we have measured by real-time PCR using primer specific to the region, that is the region where the break was induced, that is 0.18 towards the right of that region, kilobases, 2.1 kilobases towards the right of the damage site. So you can see that this is the DNA levels. So this is the 0.18R wild type, and this is the FUN30 deletion. So in FUN30 deletions, with the passage of time, the DNA level is high compared to the wild type. So which means that in the wild type, the resection is more than in the FUN30 deletion. So this means that FUN30 is favoring the DNA and resection even closer, even at the shorter range, at the short range level. All right. So in summary, we can say that several proteins that were belonging to nuclear membrane, they are pulled down along with FUN30. FUN30 is physically interacting with NSP1. FUN30 is a nuclear protein. It is also involved in short range of DNA and resection. Thank you so much for listening me. Bye-bye.
Thank you so much. This was a great presentation, Dr. Mavish, and I would like Dr. Asif to take the floor and speak few words, please. Please unmute yourself.
Sorry. Thank you, Dr. Sarah. It's really honored for speaking a few more words on behalf of our Pindi Medical University Office of Research, Innovation, and Commercialization. So just to share with all the globe and with the stakeholders that our Pindi Medical University is taking few steps that are way ahead, inshallah, towards the digitalization in Pakistan. And we are improving the quality of standard education by giving the inputs inside to medical students, the faculty member, residents, and the future doctors regarding the innovation and entrepreneurship. So this is, beginning towards a new era that is entering in the world of innovation and entrepreneurship. So we're inculcating the innovation culture in medical students, and we're also linking with the regional universities, the informatic universities, biotechnology universities, engineering universities, electronic and other computer sciences, biotechnology, biomedical industry, biomaterial industry, agriculture industry. So these all are steps towards achieving the sustainable development goals. Currently, we are very having positive steps, and in the impact ranking system, Rawalpindi Medical University is leading the region, and it is given the high ranking, first ranking in Times education system as well. So we have also developed the integrated undergrad innovation curriculum, the integrated curriculum for entrepreneurship as well. And we are sensitizing the Rawalpindi Medical University medical students, giving them the insight and sensitization, the workshop, the hands-on training for the innovation culture, so they can link with the other regional universities. This is the the vision of our Vice Chancellor, Professor Mohammad Umar Tara Imtiaz Hilal Imtiaz, which is very visionary leader, and his timely steps lead all the region in future specs. So achieving the sustainable development goals is really very quite some dream for all the region, especially for Pakistan. in this era where we are fighting against the double burden of disease. And we are also having the issues and problems, the alarming situation like community engagement, and health risk behaviors are very compromised. So we are trying to digitalize all the system and all the services for these all improvements, and leading towards also the improvement in primary healthcare services. Many medical justice also putting some of the measures so that the on the ground realities in the primary setups that can be well addressed, and we can address the community problems, preventive aspects to improve, so that the double burden of disease, in the double burden of disease, we can reduce the infectious elements, and the vaccination coverage, the maternal and child health services, as well as the reproductive and sexual health services of the women, especially women empowerment, the women education, and also the other aspects like TB, malaria, and dengue. These all the infectious diseases which we need to be well addressed and well communicated with the community. So community can take the right decisions to improve the quality adjusted life years. And we are very thankful for all the leading universities for connecting and encouraging our steps. And right now, you will be happy to hear that we are linking with the other regional universities for indigenous productions of different innovative aspects, and in genetics, in skin products, especially other the gadgets and the equipments which are really need in Pakistan, because Pakistan healthcare system is also compromised in these all missionaries where we are have to look onto the other countries for the import. And other thing which I want to share, that is China-Pakistan Medical Association, which is a well-known association. Our president is Dr. Mohammad Shahbaz, is well connected with our Penny Medical University. We are putting some of the 4 steps for improving academics, research, and innovation. And we are also connecting with the Chinese universities for the major collaborations in augmented virtual reality setups. So these are the few one belt, one health road, services which are shaping Pakistan into healthy Pakistan and prosperous Pakistan. So this is from my side, and I would request all the stakeholders, policymakers, and the think tanks, and all those who are philanthropists or who are very keen to see Pakistan innovative, prosperous Pakistan. Please come forward, help Rawalpindi Medical University, because it is public university. And being as a public university, we need your support in terms of all aspects, whether they are connecting with the big stream innovators, and in the aspect of the financial aspects are very important. We want to build the incubation center, and want to raise all the standards of the innovation. So I'm looking forward for all the stakeholders. Thank you from my side, and we together will make Pakistan prosperous, healthy Pakistan, and we'll achieve the SDGs 2030. At least if we can't achieve all, but we want to put something 90% benchmark, so that we can have our say in the globe. Thank you so much.
Thank you, Dr. Asif. Thank you for your explanation and the insightful and the future directions. It is really the need of the time where we all gather on a platform where the ideas can be shared and policies can be made. Only that could not be possible with a solo flight. We have to contribute towards each other. So, Shintaro, are you there? I would like to tell you about our last presenter, Dr. Haq Nawaz. He is having some kind of issue uploading the video, and also due to network problem, he is not able to join us.
Okay.
Okay, so I think that this is it for because we are really sorry and apologize on behalf of this networking glitch that is happening over here. Dr. Haq Nawaz unfortunately is not able to join because I have told him to send us the updated recorded video, but it's unfortunately not possible for him. So at last, I think we can call it a day. And I would like to thank all the esteemed guests, fellow presenters, and participants for your active engagement and valuable contribution in the medical working group in APAN. I would like to extend my sincere gratitude to APAN for providing a platform for the experts to share their knowledge and collaborate. Rawalpindi Medical University, specifically Professor Muhammad Umar, for giving us a great platform to present ourselves, Higher Education Commission for their support and engagement. And last but not the least, I would like to thank you, Shintaro, for mediating this session. Any concluding remarks from your side?
No, thank you very much for preparing in advance for even though we had network problems, I think everything went well. Thank you very much.
Yes, thank you so much. Thank you so much. And we look forward for in the future for more such meetings. Thank you.